Now, we have been discussing the application of ELISA system in determining the antibody immune reaction in plants, in blood, and all that. Today, I will particularly discuss in this module the enhanced ELISA system. What are they and how they are used? Now, the maximum sensitivity of ELISA is in the picomole range. You see, whenever we use any method for a diagnostic purposes, it has to be extremely sensitive. And the maximum sensitivity of ELISA, that is in the range of picomole range. Very, very considerably small range. But the physical limitations, they are based on the dynamics of the double binding events. You see, we discussed that there are the events where there is a double binding, antigen, antibody binding, antigen, enzyme, antibody binding, and like that. The subsequent generation of signal which is produced is the background substrate value as well. You see, in the diagnostic test, there are signals which are produced, and there are always background of the substrate value which is present there. So if it has to be sensitive, that signal should be strong enough to overcome or to generate a signal which should be above the background of the substrate level. That is common for all the diagnostic tests, whatever we devise. Now, antibody binding, it cannot be improved. One thing we have to keep in mind, that the binding capability of antibody to the antigen shall remain the same. It cannot be improved, as it is primarily a random event modified by the individual avidities of the antibodies themselves. That is, the antibodies which are present there, their reaction or their binding with the antigen will remain the same. It cannot be improved. Unlikely, to the chemical substrate, you can see we can increase the reaction by increasing the concentration of the components or vice versa. But one thing remains over there, that the binding of the antibody with the antigen, that cannot be improved, and it is a random event. Now, how we can improve this? There are many methods. For example, temperature modification can be done. The reaction can be carried out some temperature which is optimum. So some antibodies, they perform better at the specific temperature. Some antibodies will perform at high temperature. Some antibodies will perform at low temperature. But there will be an optimum value of the temperature at which those antibodies will perform optimally. Now, more enzyme that can be bound by using. You see, because the reaction is between the enzyme and the antibodies. Now, the more enzyme can be used by testing multivalent attachment molecule. That is, we can use different molecules. We can construct the molecules which are multivalent, and that can improve the enzyme that is the bound to the antibodies. For example, evident biotin binding system is there, and this system, it using the evident biotin binding system, it allows amplification through this route. That is, with the multivalent attachment, it is improved the attachment, and both evident and the biotin, we must keep in mind, they are the tetravalent. So they are the multivalent components which are used. The detection antibodies is then labeled with the biotin and the reporter enzyme with evident. Remember, we have discussed that we need two antibodies and the reporter enzyme which are attached and which become the marker. So what we have done, we have used, or we can use, the evident biotin system for increasing the reaction. The high affinity and the multivalency of the reagent allow larger complexes of the enzyme to be linked to the detection antibody. So if there is a larger complexes, they are made by multivalent, then that is attached with that, and it increases the linking power or the detection power of the antibody. Now, alternative amplification step is by enhancing the substrate reaction. We can increase the substrate reaction as well, which is usually by using a double enzyme system. We use the double enzyme system and the reaction is increased. Now, primary enzyme, which is bound to antigen, it catalyzes a change in the second enzyme, which is then generated, which then generate the signal which can be measured. So here again, we are using the multi-enzyme system which are bound to the antigen, and the primary enzyme, it catalyzes a change in the second enzyme, which is then generates the signal. Alkaline phosphatase, it conjugated secondary enzyme that can be used to drive a secondary reaction. So we are increasing the reaction speed, different reaction, I should say, the component for that purpose. Now, secondary reaction which involves NADP, dephosphorylation to NAD, and it further is reduced to NADH. That is, again, it reduces to NADH by alcohol dehydrogenases. So we are using another multi-enzyme system, and tetrazolium salt that is oxidized, and this in turn creates a loop in which the tetrazolium salt is oxidized as the NADH return to NAD. So this cycle goes on from NADH to NAD, NAD to NADH. And the cyclic nature of the reaction, it causes the amplification and increases the observed color development. So we saw that in this method, we have increased the sensitivity of the diagnostic test, although we know that the test or the other attachment or other with the antigen antibody attraction, we cannot increase, but we can increase it indirectly by manipulating the enzyme reaction or by manipulating the multivalent components with that.